everyone, and welcome to our first of two videos on the muscular tissue section of our muscle system. So we're going to be going through the muscular system in two different chapters. This chapter is going to be focusing primarily on the physiology of muscle, though we're not going to be going into nearly as much detail as you will in physiology. We're going to emphasize the physiology of skeletal muscle. And then in our next chapter, we'll be working on the gross anatomy, so how muscles work, muscle groups, actual muscles. But first we need to really understand what muscle is. Now in our lecture on tissues, we did very briefly introduce the muscular tissue types. We also talked about them in lab. So remember, we have three different types of muscular tissue. We have skeletal, we have cardiac, and smooth. And if you remember, they all have different properties. They all look a little bit different, but today what we're going to focus on primarily is skeletal muscle. A little bit more detail about its microscopic structure, and then of course how it works. With cardiac and smooth, we'll talk very briefly about their structure and very briefly about sort of the differences between them and skeletal muscle. We will not be getting into how they contract or anything like that, because it is very, very weird, I guess would be the best way to say it. Now, skeletal muscle, if you remember, it is striated. So we have these sort of vertical bands, these alternating dark light bands. We'll talk about how we get those soon. It is voluntary, which means this particular tissue is under conscious control. I can tell my muscles when to contract and when to relax. There are some that we don't think about. So the ones that you're using right now to keep your back upright while you're watching this video, you're not consciously telling your abdominal muscles or your back muscles to contract at the right tension so that you can maintain posture. They just happen. Same thing with your diaphragm. We can consciously control the rate of our breathing, like you do in yoga or when you're doing meditation, but most of the time we're not consciously telling our diaphragm to contract and relax every time we take a breath. So that's also under subconscious control. Another thing about skeletal muscle is it's multinucleate. So you can see a bunch of nuclei inside the skeletal muscle. Cardiac muscle is also striated, so we saw that in lab. It has these vertical alternating dark light bands. However, it is involuntary, so that's one thing that makes it different from skeletal muscle tissue. We don't tell our heart muscle to contract. It's actually what we call autorhythmic. So it sets its own rhythm, and we'll talk more about that when we get to the heart in a few videos. Now, when we look at the structure of a cardiac muscle cell, remember it's a little bit different from skeletal in the fact that, remember, skeletal was cylindrical, so when we looked at it under the microscope, it looked like a rectangle. When we look at cardiac muscle, it is branched or bifurcated. So we see these branched cells. That's why you see a lot of this empty space here. It's filled with connective tissue. So we have these branched cardiac cells, and you'll also see we have these dark vertical bands. Remember, these are called intercalated discs, and if you recall from our first unit, these are gap junctions. Let's just figure that one out again. Junctions. So remember, these are the little types of tissue connectors. They're made of connexons. They allow ions to move through really quickly. And as you'll learn in physiology, this is really important for getting the heart muscle to contract as one unit. Now, lastly, we have our smooth muscle tissue. And as you can see, there are no stripes, no striations. It's not that they don't have the same contractile elements. They do. Same ones as skeletal and cardiac. The major difference is they're oriented differently, so that's why we don't see those stripes. Now, just like cardiac muscle, smooth muscle is involuntary. A lot of times it will be autorhythmic, so when we think about the gut, the process of moving food through your gut, this is an autorhythmic process. It has this basic rhythm to it, and you guys will learn more about that in physiology. Where do we find smooth muscle? The easiest way to think about it is we find it in the walls of hollow organs, because we haven't really gone through our organ systems yet. It's kind of hard to pinpoint exactly where they are, but think of anything that's hollow: a blood vessel, the GI tract, the uterus. Those are all places that are going to have smooth muscle. We also have them in our eye, as we learned when we talked about the integumentary system. We have them in our skin, controlling our hairs. Now, when we look at muscular tissue as a whole, there's different functions to it, right? And one of the major ones we think about is movement. So we typically think of body movements, walking, moving our arms, and things like that. But we also can think about it in terms of movement of substances. So when you think about the heart, it's pumping to move blood. The GI tract is moving foodstuffs through its different regions. We also use our muscular tissue to stabilize body positions, basically postural muscles. And then, of course, we use muscle to generate heat. So when you get cold, when your body temperature drops, in order to maintain homeostasis, to try to bring that temperature back up, you start involuntary muscle contractions of your skeletal muscle. And that's what gives you shivering, because we're using a lot of chemical reactions to create the energy to make you shiver. You're going to create heat. Now, muscle tissue also has 4 basic properties to it. We have excitability, contractility, extensibility, and elasticity. So excitability, that means that it's able to respond to a stimulus, some sort of a change or a trigger, okay? And it can produce action potential. So as we'll see a little bit at the end of this lecture and then into the next one, one of the ways we tell our muscle to contract, the way we do that is with an action potential, a electrical signal. When the skeletal muscle or the cardiac muscle or the smooth muscle gets that electrical stimulus, it's going to lead to what's called a contraction. So when the muscle is told to contract, it's going to shorten, and what happens is it generates force or tension, and that's what creates the movement. Now, with the ability to contract, we need to have a couple other properties: extensibility and elasticity. Elasticity is the easiest one to talk about because we discussed this when we talked about our tissues, right? Elastic connective tissue, elastic cartilage, right? They have these elastic fibers that are allow the tissue to stretch and recoil. So with your muscle, when it contracts, it recoils back to its original shape. Extensibility is the ability of the muscle to stretch without being damaged. So when you are doing different yoga postures or you're just stretching in general, you're not typically tearing the muscle. Your heart also stretches as well. As it fills with blood, it can stretch, but it's not going to tear or damage the muscle. Once again, there's always caveats to this, especially with skeletal muscle. Now, skeletal muscle is going to be the major focus of this particular chapter, once again, because that's what we're looking at in the next chapter. Skeletal muscle is made up of a bunch of different muscle cells that will bundle together that then create the whole muscle that we're going to be learning in our next chapter. Keeping everything together is a series of connective tissues, and they all have different names. So we have epi, peri, and endomysium. So we have endo, peri, and epi. So when we look at these names, we can kind of think of our root words. Mysium, that's referring to the muscle. Epi is sort of outside of or surrounding. And that's the outer layer of our muscle. Peri sounds like perimeter. So this is going to be sort of around a bundle of muscle cells. Okay, so this would be like surrounding on the outside. This would be sort of just around a grouping. And endo means within. So that's going to be our innermost layer. Now what the perimysium is doing right here, it is going to be bundling all of the muscle cells. And that bundle of muscle cells is something we call a fascicle. And these fascicles are held together by perimysium. And we'll talk more in the next chapter about the importance of these fascicles. They're what kind of give you the muscle lines that you see when we do dissections and things like that. And it's the orientation of these fascicles that provide different properties to each muscle. And wrapping each individual muscle cell or muscle fiber or myocyte is a layer called the endomysium. And all three of these layers of connective tissue are all going to fuse at either end of the muscle where it attaches at the bone with something we call a tendon. So a tendon attaches muscle to bone, and ligaments attach bones to bones. So that's what we looked at in the end of our last unit when we talked about things like the knee. Tendons are attaching muscles to the bone. Now, tendons are like a big rope, whereas another type of muscular attachment we have is called an aponeurosis. And an aponeurosis is essentially if you took that tendon and flattened it out with a rolling pin, that's an aponeurosis. We have them on our abdomen. There's one on the top of our head. So they're very important. They're just flat tendons, essentially. Now, one other thing that we have going on with muscle is something called fascia. Now, fascia is going to be important because it helps, in the case of our class, the type we're looking at, is it's going to be separating muscle groups. So it's just tissue that we find in between muscles, but we can find it a lot of different places. We can find it not only around muscles, but in other places of the body, and it has different roles. It helps with sort of compartmentalizing different areas. It helps with reducing friction or bringing blood back to the heart. There's a lot of different roles for fascia, and there's a lot of different ways to categorize it. But as with a lot of things we will see in anatomy, we just don't have a fully agreed way to name the fascia. So that's why we're not going to be learning all the different types. Now you can see a couple examples here. This is something you would see when you're doing a dissection, that sort of fibrous, almost spiderweb-like material. This is what we see as we start to separate muscle groups, like in the forearm. You can see this white tissue. It separates very easily. That's fascia. A real-life example, when you peel the skin off of a chicken breast, you can see this sort of connecting fibers. That's fascia. That's connecting the skin, underlying fat to the muscle. So that would be fascia. So why are we talking about this? Many of you have probably partaken in this, something called myofascial release. This is something people use in physical therapy or just in their daily lives to help reduce pain or reduce the sort of knots, I guess you could say, that are reducing their range of motion. Now, you can do things like you see here, where you lay on like the hard tennis ball, the roll, like the massage rolls that you have. Those are also a type of product you can use for myofascial release. But why are we talking about it? It's because there's not been a lot of really good scientific studies that show definitively that it works. Some studies have shown that there were positive outcomes, but there weren't a lot of really good studies. So when we look at studies in science, we want to see a lot of numbers. We want to see a high number of test subjects, because the more numbers we have, the better our data will be. And you guys will learn this in statistics. The higher N value, the more likely you're to get that nice standard curve. And it won't skew the data one way or another. Typically we want to at least an N of 3, but we want a lot more. We want to have a really, really good overall result. So these particular studies showed that there weren't a lot of test subjects. So that means the data could have been skewed one way or the other. Another review, which is where they look at a bunch of different studies, they don't actually perform the experiment themselves, they show that there's a lot of anecdotal evidence, but not really a lot of experimental evidence. So anecdotal would be someone saying, yes, I do it and it makes me feel better. So not really testing it out or, getting some sort of measurable result. So if it works for you, that's awesome, but there's not a lot of actual real scientific evidence that I could find at the time of writing this lecture that was definitive experimental. Now with muscles, okay, they are tissue, so they need blood supply in order to work. And in order to be told what to do, they need a nerve supply. So we aren't going to get into neurons and nervous tissue till the end of the semester, but what we're looking at here is a motor neuron. This is coming from your brain, and it's going to tell your skeletal muscle when to contract. And this part here would be in the brain. This is what we call the axon that travels from the brain, and you can see at the end it branches, and we call these axon terminals. These will be important at the end of today's video because we're going to talk about how we actually get that signal to the muscle. And in terms of blood supply, each muscle cell is going to have a lot of capillaries nearby it. And the reason for that is, so you can see all the capillaries here, is because muscle requires a lot of oxygen in order to make its ATP. And we need to make a lot of ATP because, as you'll see in physiology, we start off with maybe 6 seconds of energy. So we need to be able to continuously make that energy. And in order to do that, I need sugar and I need oxygen. And I get all of that from the blood. On the left here, you can see individual muscle cells, and then these are those little ends of the neurons called the axon terminals. So one neuron can actually control either just a handful of muscle cells, or they can control a huge amount of muscle cells. And that will let us know, the types of contractions we can get. They could be fine motor if they're just a few, because they can control very specific actions, or if it's a postural muscle, we'll probably be controlling a huge number of muscle cells. Now skeletal muscle, I like to describe its structure as a Russian doll. It's basically cylinders, inside cylinders, inside cylinders. So we have our muscle, and then we end up with our fascicle, which, remember, this is just a bundle of muscle cells. And then we have a muscle cell, or a myofiber, or another name for it is a myocyte. Note that root word myo is one of many that we use for muscle. Inside a muscle cell, we have a bunch of cylindrical organelles called myofibrils. This is just a type of organelle. And each myofibril, which you can see here, and we're going to take a look at later, is made of these contractile elements called sarcomeres. And this is what we call the functional unit of muscle. So the functional unit of any organ is the part that's actually doing the job of that particular organ. In this case, skeletal muscle, it's to contract. So where we're getting that contraction from is from the sarcomere. And the sarcomere is built, and we'll look at this too, of what we call myofilaments. Remember, these are cytoskeletal elements, and these would be actin and myosin. And their arrangement creates a sarcomere, okay? So what we're going to do is we're going to take a closer look at this arrangement, because without understanding the arrangement and the other structures we see on like the myofibrils, we're not really going to understand how a muscle contracts. So first thing first, if we look at a muscle cell, it's going to have a plasma membrane. It's a cell. And the fancy name for a plasma membrane of a muscle cell is a sarcolemma. Okay, so sarco, you're going to see as the root word a lot for some of these structures. It's also the name of my cat, so you can remember it that way too. So we can see the sarcolemma, which is going to be this blue area here. Now, one of the issues, if you think about it, you're going to build a muscle cell, is I have a 3-dimensional cylinder. And if my electrical signal is only going on the outside here of the plasma membrane, only the outer only the outer organelles are going to be told it's time to contract. So in order to get deep, to get the deeper organelles, the contractile elements cued that we're going to be contracting, we're going to have these invaginations, or the inward dipping of the plasma membrane. And that's what you see here and here and here. It's these sort of light blue tubes. Those are called transverse tubules, or T-tubules for short. They're going to help get the signal deeper into the cell. And inside the cell, because it is a cell, we have cytoplasm, but because it's a muscle cell, we call it sarcoplasm. And it has a different composition. Because it's a muscle cell, it requires different ratios of molecules. So it's really high in mitochondria floating around in here because they make the energy. And in order to do that, right, I said we need sugar and we need oxygen. Well, one of the ways we get sugar is with glycogen. Glycogen is just long chains of glucose. And we'll talk more about how we get that glycogen into the skeletal muscle cell a little bit when we talk about the endocrine system. And once again, you'll talk way more about it in physiology. So we kind of store some sugar. And then myoglobin, this is a protein that's similar to what we'll talk about more in blood called hemoglobin. And it's a protein that binds oxygen. So that's how we get our oxygen, myoglobin. Now we also have, right, again, I told you we have our myofibrils, which are these guys here. And on the myofibrils, we have a couple of, well, we have a really important structure. It's this weird beige netting that you see called the sarcoplasmic reticulum. It's a modified smooth endoplasmic reticulum whose job is to store calcium. So how does it know to release the calcium? Well, we have these T tubules, remember? This is where the electrical signal's coming through. And the T tubules abut next to the edges of the sarcoplasmic reticulum that we call terminal cisternae. Okay, whenever you see AE as the end, that means it's plural, so there's multiple. Now these terminal cisternae come together and meet up with the transverse tubule. So we would have something like this. We'd have, here's our two terminal cisterns, and then in the middle is the T tubule. So there's the T tubule, here's the parts of the SR, our sarcoplasmic reticulum. So as the, and you'll hear me say this a lot, but as the electrical signal comes down the T tubule, it's going to tell the sarcoplasmic reticulum to open up and release calcium, which will be important momentarily. We then, if we look at the sarcomere, we can see all these lines here. These are the actin and myosin arrangements. And that's what we want to take a look at next. The arrangement of those myofilaments, the actin and myosin, that's what gives us our dark light bands. So the edge of each sarcomere, which you can imagine sarcomeres, they're just lined up next to each other from one end of the muscle to the other. So you'd have a bunch of them lined up. And each end is called a Z disc, because if you look at it, it kind of looks like, you know, Z's that you just keep writing, little zigzags. The Z discs are attached to our actin, okay? And then running down the middle, we have our thicker filament, and that's going to be myosin. Now, where myosin and actin overlap, that's called the A band. So this whole area here is the A band, because even though we don't see overlap here in the middle, there will be overlap when we do a contraction. In the middle, we have a protein that's going to basically show us the midline of the sarcomere, and that's actually where actin is going to move to. So actin will, when we have a contraction, move in this direction, shortening the sarcomere and therefore the muscle. Now the areas that we have that have no overlap of actin and myosin, they're just actin only, this is called the I band. So the I band is just actin, the A band is actin and myosin, and when we have a relaxed muscle cell, we have an area where there's no overlap yet, here in the middle, and that's called the H zone. So the H zone is just myosin where we aren't going to have, when we don't have a contraction, okay? So we have M line, H zone, A band, I band. And what's going to happen is when we contract the muscle, those actin and myosin are going to slide over each other, shorten the sarcomere, and we're going to get a contraction. And that's the whole focus of our next lecture, so I won't get into it too much here. But in order to be able to talk to you guys about the whole process of a contraction, we need to look at all the different components that could be involved. Now we have a lot of different proteins in skeletal muscle. Some of them are contractile, so they're going to be shortening the muscle cell. Some of them are regulatory, so that means they're going to help turn the contraction on or off. And then others are just there for structure, just to make sure that everything holds together well. So let's look through these guys. First up are the contractile proteins. These are actin and myosin. So actin, once again, is the thin one, and myosin is the thick one. Myosin has two binding sites for itself, one for ATP, our energy, and one for actin. Actin is going to have binding sites just for our myosin heads. So they're going to bind up, come together, and that's how we're going to shorten our muscle cell. Now, how do we make sure we contract when we want to and we don't contract when we don't want to? What you're looking at here, the yellow dots, these are the proteins of actin. And you'll notice running along them, covering the binding sites for myosin, is this little white strand. And on the white strand, we have a little complex of purple proteins. So the white strand that you see, this is called tropomyosin. Tropomyosin is going to block the binding sites for myosin. Myosin is always ready to bind to actin, but we don't always want it to do that, right? I don't want my muscles in a constant state of contraction or tetany. I want them to relax at certain points. So as long as tropomyosin is blocking the actin binding sites, we cannot have an actual contraction. On the tropomyosin is a series of 3 proteins that we call the troponin complex. Now, the one in the middle, this one is going to bind to calcium. So troponin binds to calcium. And one thing that's really important in terms of biochemistry is whenever something binds to or unbinds from a protein, we're going to get a change in shape. So when calcium, which are these little green dots here, binds to troponin, it's going to move the whole troponin-tropomyosin complex. So now you can see the myosin binding sites are exposed. And where are we getting that calcium from? The sarcoplasmic reticulum. And when do we get that released? When there's an action potential. So that'll lead to the release of calcium. It's going to bind troponin, move tropomyosin, and now we can get a shortening of the sarcomere. Now, in terms of our structural proteins, we have 3 that we want to talk about: titin, nebulin, and dystrophin. So titin, if you look back at the sarcomere that we were looking at, there were attached to the myosin heads these little curlicues. They look like springs attached to the myosin filament, I should say. Those are titin. They are like little springs. So what happens is the myosin is going to move the actin over, and that's going to kind of compress the springs, and then when we want to relax, they jut outward. So this is what gives us that elasticity and extensibility property of our skeletal muscle. Nebulin is going to anchor the thin filaments to the Z disc. So you guys can see the Z discs here, and there would be little proteins attaching the thin filament here to the Z disc. Those are going to be our nebulin. Okay, so these little dots here would be nebulin. So that's how your actin binds to the Z disc. Now, dystrophin, as the name sounds, like dystrophy, this is the protein that is damaged, missing, dysfunctional in Duchenne's muscular dystrophy. So DMD is Duchenne's muscular dystrophy. What dystrophin does is it helps connect the actin to the sarcolemma. And if we don't have that dystrophin, it's going to destabilize the muscle cell and it won't be able to contract properly. So that's why you get those symptoms of Duchenne muscular dystrophy, the atrophy and things like that. So dystrophin is the protein that is causing those issues. Now, what we're going to be getting into in the next video is something called the sliding filament mechanism or model. And what this states is that when the muscle is stimulated, the filaments, actin and myosin, will slide over one another towards end line, shortening, generating force, and creating a muscle contraction. So what we're going to be talking about in the next video is how that whole process works. But what we're going to end with is how we actually get it started. Okay, so we're going to talk about how do we trigger the event, and then in the next video, guys, we'll run through the entire process from start to finish. So one final structure that we haven't talked about is something called the neuromuscular junction. I'll draw you guys a picture in just a minute. The neuromuscular junction, as the name sort of indicates, neuro means nervous or the brain, and muscular means muscle. So this is basically where the neuron meets the muscle plasma membrane. With neural connections, it's important to understand that they don't physically touch. So neurons don't physically touch other neurons. They don't touch their effector cells, whatever they're trying to stimulate. What they create is something called a synapse and a synaptic cleft. So this whole region here, this would be a synapse. It's a connection point between, in this case, two neurons. The area where they don't touch is the synaptic cleft. And this is important because, as you'll learn more about in physiology, and we'll talk a little bit about today, is the way we signal from neuron to neuron or neuron to effector is through the diffusion of what are called neurotransmitters. So neurotransmitters are just chemicals that are released from neurons that tell other neurons or their effector cells what to do, stimulate or inhibit. And there's lots of different neurotransmitters that have lots of different roles. And the one that we're concerned with in this course is called acetylcholine. So acetylcholine, or ACH, will be released from the somatic motor neuron that's controlling that muscle cell. And it diffuses across the synaptic cleft, okay, to the muscle cell, binding to a receptor, triggering the action potential that leads to our muscle contraction. Now, where we have those receptors is a region called the motor end plate. So I kind of want to draw this out for you guys. So here is the end of our neuron. And then we have our little motor end plate. Let me erase that. That doesn't look very good. Let's try this again. Okay. So we have this. We have a motor end plate. Continuation of the sarcolemma. So what I'm going to draw you guys is kind of just the basic idea of what we're going to see in the next video. Okay, so what we have is sort of a blown up view of the neuromuscular junction connected to the rest of the system here. So let's kind of label everything. So here is the axon terminal. And inside of it, what we have, these little green dots, that's acetylcholine. So we're going to get an electrical signal that's going to go down. I'll draw it as yellow. It's going to come down from the brain, down the axon terminal. And it's going to tell acetylcholine to get released. And acetylcholine will get released into the synaptic cleft and bind to these little receptors here, okay? And when acetylcholine binds to these receptors, oh my gosh, it's going to create a signal. Oh goodness, this tablet. There we go. Now, where these receptors are located, so this is an ACH receptor, they're on a specific part of the sarcolemma that we call the motor end plate. And the reason we call it this, the reason it's so specific, is this is the area right underneath the axon terminal. I don't want acetylcholine receptors everywhere along the sarcolemma because I'm not getting directly stimulated along the entire sarcolemma. I'm only getting stimulated by acetylcholine underneath that axon terminal. So it only makes sense to have them there. So when the acetylcholine binds to the receptors, it's going to trigger another action potential. okay? And so that action potential is then going to run along and down the sarcolemma. So what is this called? Let's go back to our anatomy here. So here is the sarcolemma. Oh goodness. Okay, so this is our sarcolemma here. This is that invagination, the T tubule. This is our sarcoplasmic reticulum, the SR, and it's full of calcium. Now I'm going to zoom in on this area here. So here's the T tubule meeting up with the terminal cistern of the SR. What we have on here are little receptors. You'll get more into them in physiology. But these receptors are what we call voltage gated. So they're voltage gated calcium channels, not receptors, excuse me. Voltage gated means that these channels are not going to open unless there's an electrical signal. So as that electrical signal moves down the T tubule, it will cause these channels to open and release calcium. Okay, and then that's going to lead us into what we're going to see in the next video. So here's a very amazing diagram showing you the neuromuscular junction. We have our action potential in these little vesicles. We have acetylcholine, and you can see the receptors here. It goes into more detail in terms of physiology of like the movement of sodium and potassium, the influx of calcium that leads to the release of the acetylcholine. That's stuff that we don't need to know in this class. But those of you that are also taking physiology, this might be a good figure to go back to when you're learning about your skeletal muscle physiology. So that's it for this part of our lecture on muscle physiology. In the next one, you're going to go through the whole process of muscle contraction, and we'll talk about some clinical connections as well. Okay, see you guys then.